Chapter 4 Animal Kingdom. When you look around, you will observe different animals with different structures and forms. As over a million species of animals have been described till now, the need for classification becomes all the more important. The classification also helps in assigning a systematic position to newly described species. 4.1 Basis of classification. In spite of differences in structure and form of different animals, there are fundamental features common to various individuals in relation to the arrangement of cells, body symmetry, nature of coelom, pattern of digestive, circulatory or reproductive systems. These features are used as the basis of animal classification and some of them are discussed here. 4.1.1 Levels of organizations. Though all members of Animalia are multicellular, all of them do not exhibit the same pattern of organization of cells. For example, in sponges, the cells are arranged as loose cell aggregate, that is, they exhibit cellular level of organization. Some divisions of labor activities occur among the cells. In coelentrates, the arrangement of cells is more complex. Here, the cells performing the same functions are arranged into tissue, hence is called tissue level of organization. A still higher level of organization, that is organ level, is exhibited by members of Platyhelminthes and other higher phyla where tissues are grouped together to form organ, each specialized for a particular function. In animal like Annelida, Arthropoda, molluscs, Echinodermata and chordates, organ have associated to form functional systems, each system concerned with the specific physiological functions. This pattern is called organ system level of organization. Organ system in different groups of animals exhibit various patterns of complexities. For example, the digestive system in Platyhelminthes has only a single opening to the outside of the body that serves as both mouth and anus and is hence called incomplete. A complete digestive system has two openings, mouth and anus. Similarly, the circulatory system may be of two types. One, open type, in which the blood is pumped out of the heart and the cells and tissues are directly bathed in it, and second, closed type, in which the blood is circulated through a series of vessels of varying diameters arteries, veins and capillaries. 4.1.2 Symmetry. Animals can be categorized on the basis of their symmetry. Sponges are mostly asymmetrical, that is, any plane that passes through the center does not divide them into equal halves. When any plane passing through the center axis of the body divides the organism into two identical halves, it is called radial symmetry. Coelentrates, ctenophores and echinoderms have this kind of body plan. In figure 4.1a, animals like annelids, arthropods, etc., where the body can be divided into identical left and right halves in only one plane, exhibit bilateral symmetry. In figure 4.1b. Figure 4.1a radial symmetry, figure 4.1b bilateral symmetry, figure 4.2 showing germinal layers, A diploblastic, B triploblastic. This is diagram of diploblastic. This is outer ectoderm, inner endoderm. This is mesoglea, and this diagram of triploblastic, outer ectoderm, inner endoderm, and middle is mesoderm. 4.1.3 Diploblastic and triploblastic organization. Animals in which the cells are arranged in two embryonic layers, an external ectoderm and internal endoderm, are called diploblastic animals. Example, coelentrates and undifferentiated layer mesoglea is present in between the ectoderm and the endoderm. In bracket figure 4.2a, those animals in which the developing embryo has a third germinal layer, mesoderm, in between the ectoderm and endoderm are called triploblastic animals. In bracket platyhelminthes 2 chordates. Figure 4.2b. 4.1.4 Coelom. Presence or absence of a cavity between the body wall and the gut wall is very important in classification. The body cavity, which is lined by mesoderm, is called coelom. Animals possessing coelom are called coelomates. Example: annelids, molluscs, arthropods, echinoderms, hemichordates and chordates. In bracket figure 4.3a, in some animals the body cavity is not lined by mesoderm. Instead, the mesoderm is present as scattered pouches in between the ectoderm and endoderm. Such a body cavity is called pseudocoelom and the animals possessing them are called pseudocoelomates. Example: Aschelhelminthes. In bracket figure 4.3b, the animals in which the body cavity is absent are called acoelomates. Example: Platyhelminthes. In bracket figure 4.3c. Figure 4.3 diagrammatic sectional view of A. Coelomate, B. Pseudocoelomate and C. Acoelomate. 4.1.5 segmentation. In some animals, the body is externally and internally divided into segments with a serial repetition of at least some organs. For example, in earthworm, the body shows this pattern called metameric segmentation and the phenomena is known as metamerism. 4.1.6 Notochord. Notochord is a mesodermally derived rod-like structure formed on the dorsal side during embryonic development in some animals. Animals with notochord are called chordates and those animals which do not form this structure are called non-chordates. Example: Porifera to echinoderms. 4.2 Classification of animals. The broad classification of Animalia based on common fundamental features as mentioned in the preceding section is given in figure 4.4. Table: Kingdom Animalia in bracket multicellular. Phylum Porifera. Level of organization: cellular level. Symmetry: mostly asymmetrical. Body cavity or coelom: acoelomate. Coelenterata in bracket Cnidaria. Level of organization: tissue, organ system. Symmetry: radial. Body cavity or coelom: acoelomate. Phylum Ctenophora. Level of organization: tissue, organ system. Symmetry: radial. Body cavity or coelom: acoelomate. Phylum Platyhelminthes. Level of organization: tissue, organ system. Symmetry: bilateral. Body cavity or coelom without body cavity in bracket acoelomate. Phylum Aschelhelminthes. Level of organization: tissue, organ system. Symmetry: bilateral. Body cavity or coelom with false coelom in bracket pseudocoelomates. Phylum Annelida. Level of organization: tissue, organ system. Symmetry: bilateral. Body cavity or coelom with true coelom in bracket coelomates. Phylum Arthropoda. Level of organization: tissue, organ system. Symmetry: bilateral. Body cavity or coelom with true coelom in bracket coelomates. Phylum Mollusca, Echinodermata, Hemichordata and Chordata. Level of organization: tissue, organ system. Symmetry: bilateral. Body cavity or coelom with true coelom in bracket coelomates. Echinodermata exhibits radial or bilateral symmetry depending on the stage. Figure 4.4 Broad classification of kingdom Animalia based on common fundamental features. The important characteristic features of the different phyla are described. 4.2.1 Phylum Porifera. Members of this phylum are commonly known as sponges. They are generally marine and mostly asymmetrical animals. In bracket figure 4.5. These are primitive multicellular animals and have cellular level of organization. Sponges have a water transport or canal system. Water enters through minute pores in bracket ostia in the body wall into a central cavity, spongocele, from where it goes out through the osculum. This pathway of water transport is helpful in food gathering, respiratory exchange and removal of waste. Conocytes or collar cells line the spongocele and the canal. Digestion is intracellular. The body is supported by a skeletal made up of spicules or spongin fibers. Sexes are not separate. In bracket hermaphrodite, that is, eggs and sperms are produced by the same individual. Sponges reproduce asexually by fragmentation and sexually by formation of gametes. Fertilization is internal and development is indirect, having a larval stage which is morphologically distinct from the adult. Figure 4.5. Examples of porifera: A. Cycon, B. Spongia, C. Spongilla. Examples: Cycon in bracket Scypha, Spongilla in bracket freshwater sponge and Euspongia in bracket bath sponge. 4.2.2 Phylum Coelenterata in bracket Cnidaria. They are aquatic, mostly marine, sessile or free swimming, readily symmetrical animals. In bracket figure 4.6, the name Cnidaria is derived from the cnidoblast or cnidocytes in bracket which contain the stinging capsules or nematocysts present on the tentacles and the body. The cnidoblasts are used for encourage, defense and for the capture of prey. In bracket figure 4.7, cnidarians exhibit tissue level of organization and are diploblastic. They have a central gastrovascular cavity with a single opening, mouth on hypostome. Digestion is extracellular and intracellular. Some of the cnidarians, example, corals, have a skeletal composed of calcium carbonate. Cnidarians exhibit two basic body forms called polyp and medusa. In bracket figure 4.6, the former is a sessile and cylindrical form like Hydra, Adamsia, etc., whereas the latter is umbrella shaped and free swimming like Aurelia or jellyfish. Those cnidarians which exhibit in both forms exhibit alternation of generation in bracket metagenesis, that is, polyps produce medusae asexually and medusae forms the polyps sexually. In bracket example Obelia. Figure 4.6, examples of Coelenterata indicating outline of their body form: A. Aurelia in bracket medusa, B. Adamsia in bracket polyp. Figure 4.7, diagrammatic view of cnidoblast. Example: Physalia in bracket Portug, man of war, Adamsia in bracket sea anemone, Panutilla in bracket sea pen, Gorgonia in bracket sea fan and Maindrena in bracket brain coral. 4.2.3 Phylum Ctenophora. Ctenophores, commonly known as sea walnuts or comb jellies, are exclusively marine, readily symmetrical, diploblastic organisms with tissue level of organization. The body bears 8 external rows of ciliated comb plates which help in locomotion. In bracket figure 4.8, digestion is both extracellular and intracellular. Bioluminescence in bracket, the property of a living organism to emit light, is well marked in ctenophores. Sexes are not separate. Reproduction takes place only by sexual means. Fertilization is external with indirect development. Example: Pleurobrachia and Ctenoplana. Figure 4.8 example of Ctenophora in bracket pleurobrachia. 4.2.4 phylum Platyhelminthes. They have dorso-ventrally flattened body, hence are called flatworms. In bracket figure 4.9. These are mostly endoparasites found in animals indicating human beings. Flatworms are bilaterally symmetrical triploblastic and acoelomate animals with organ level of organization. Hooks and suckers are present in the parasitic forms. Some of them absorb nutrition from the host directly through their body surface. Specialized cells called flame cells help in osmoregulation and excretion. Sexes are not separate. Fertilization is internal and development is through many larval stages. Some members like Planaria possess high regeneration capacity. Example, Taenia in bracket tapeworm, Fasciola in bracket liverflux. Figure 4.9 example of Platyhelminthes. A. Tapeworm. B. Liverflux. 4.2.5 phylum Aschelhelminthes. Body of the Aschelhelminthes is circular in cross section, hence the name roundworms. In bracket figure 4.10. They may be free living, aquatic and terrestrial or parasitic in plants and animals. Roundworms have organ system level of body organization. They are bilaterally symmetrical, triploblastic and pseudocoelomate animals. Alimentary canal is complete with a well-developed muscular pharynx. An excretory tube removes body waste from the body cavity through the excretory pore. Sexes are separate in bracket dioecious, that is male and female are distinct. Often female are longer than males. Fertilization is internal and development may be direct. In bracket the young ones resemble the adult or indirect. Example, Ascaris in bracket roundworm, Bucheria in bracket filaria worm and Xylostoma in bracket hookworm. Figure 4.10 example of Aschelhelminthes roundworm. This is diagram of male and this is diagram of female. 4.2.6 phylum Annelida. They may be aquatic in bracket marine and freshwater or terrestrial, free living and sometimes parasitic. They exhibit organ system level of body organization and bilateral symmetry. They are triploblastic, metamerically segmented and coelomate animals. Their body surface is distinctly marked out into segments or metamers and hence the phylum name Annelida. In bracket Latin annulus little ring. In bracket figure 4.11. They possess longitudinal and circular muscles which help in locomotion. Aquatic annelids like Nereis possess lateral appendages, parapodia, which help in swimming. A closed circulatory system is present. Nephridia in bracket singular nephridium help in osmoregulation and excretion. Neural system consists of paired ganglia in bracket single ganglion connected by lateral nerves to a double ventral nerve cord. Nereis, an aquatic form, is dioecious but earthworm and leeches are monoecious. Reproduction is sexual. Example, Nereis ferritima in bracket earthworm and Herodinaria in bracket blood sucking leech. Figure 4.11 example of Annelida. A. Nereis, B. Herodinaria. 4.2.7 phylum Arthropoda. This is the largest phylum of Animalia which includes insects. Over 2/3 of all named species on earth are arthropods. In bracket figure 4.12, they have organ system level of organization. They are bilaterally symmetrical, triploblastic, segmented and coelomate animals. The body of arthropod is covered by chitinous exoskeleton. The body consists of head, thorax and abdomen. They have jointed appendage in bracket arthros joint, poda appendages Respiratory organs are gills, book lungs or tracheal system. Circulatory system is of open type. Sensory organ like antennae, eyes in bracket compound and simple. Statocyst or balancing organ are present. Excretion takes place through Malpighian tubules. They are mostly dioecious. Fertilization is usually internal. They are mostly oviparous. Development may be direct or indirect. Example, economically important insect. Apis in bracket honeybee, bombyx in bracket silkworm, Lucifer in bracket lag insects. Vector, Anopheles, Culex and Aedes in bracket mosquitoes. Gregarious pest, Locusta in bracket locust. Living fossils, limbless in bracket king crab. Figure 4.12. Example of arthropoda. A. Locust, B. Butterfly, C. Scorpion, D. Prawn. 4.2.8 Phylum Mollusca. This is the second largest animal phylum. In bracket figure 4.13 molluscs are terrestrial or aquatic in bracket marine or freshwater, having an organ system level of organization. They are bilaterally symmetrical, triploblastic and coelomate animals. Body is covered by a calcareous cell and is unsegmented with a distinct head, muscular foot and visceral hump. A soft and spongy layer of skin forms a mantle over the visceral hump. The space between the hump and the mantle is called the mantle cavity, in which feather-like gills are present. They have respiratory and excretory functions. The anterior head region has sensory tentacles. The mouth contains a file-like rasping organs for feeding called radula. They are usually dioecious and oviparous with indirect development. Example, Pila in bracket appelisnal, Pinctada in bracket Pierre Auster, Sepia in bracket cuttlefish, Loligo in bracket squid, Octopus in bracket devilfish, Aplasia in bracket sea hare, Dentillum in bracket tusk shell, Chitopleura in bracket chiton. Figure 4.13 Examples of mollusca. A. Pila, B. Octopus. 4.2.9 Phylum Echinodermata. These animals have an endoskeleton of calcareous ossicles and hence the name Echinodermata in bracket spiny bodies. Figure 4.14 All are marine with organ system level of organization. The adult echinoderms are radially symmetrical but larvae are bilaterally symmetrical. They are triploblastic and coelomate animals. Digestive system is complete with mouth on the lower in bracket ventral side and anus on the upper in bracket dorsal side. The most distinctive features of the echinoderms is the presence of water vascular system, which helps in locomotion, capture and transport of food and respiration. An excretory system is absent. Sexes are separate. Reproduction is sexual. Fertilization is usually external. Development is indirect with free swimming larvae. Example, Asteris in bracket starfish, Echinus in bracket sea urchin. Example, Asteris in bracket starfish, Echinus in bracket sea urchin, Antedon in bracket sea lily, Cucumeria in bracket sea cucumber and Ophiura in bracket brittle star. Figure 4.14 Examples of Echinodermata. A. Asteris, B. Ophiura. 4.2.10 Phylum Hemichordata. Hemichordata was earlier considered as a subphylum under phylum Chordata, but now it is placed as a separate phylum under non-chordata. Hemichordates have a rudimentary structures in the collar region called stomochord, a structure similar to notochord. This phylum consists of a small group of worm-like marine animals with organ system level of organization. They are bilaterally symmetrical, triploblastic and coelomate animals. The body is cylindrical and is composed of an anterior Proboscis, a collar and a long trunk. In bracket, figure 4.15. Circulatory system is of open type. Respiration takes place through gills. Excretory organ is proboscis gland. Sexes are separate. Fertilization is external. Development is indirect. Example, balanoglossus and saccoglossus. Figure 4.15, balanoglossus. This is diagram of balanoglossus. This is proboscis, this is collar, this is trunk. 4.2.11, phylum Chordata. Animals belonging to phylum Chordata are fundamentally characterized by the presence of a notochord, a dorsal hollow nerve cord, and paired pharyngeal gill slits. In bracket, figure 4.16. These are bilaterally symmetrical, triploblastic coelomate with organ system level of organization. They possess a post-anal tail and a closed circulatory system. Table 4.1 represent a comparison of salient features of chordates and non-chordates. Figure 4.16, chordata characteristics. This is nerve cord and this is notochord, this gill slits and this is post-anal part. Table 4.1 comparison of chordates and non-chordates. Chordates, notochord present, non-chordates, notochord absent. Chordates, central nervous system is dorsal, hollow and single. Non-chordates, central nervous system is ventral, solid and double. Chordates, pharynx perforated by gill slits. Non-chordates, gill slits are absent. Chordates, heart is ventral. Non-chordates, heart is dorsal if present. Chordates, a post-anal part in bracket tail is present. Non-chordates, post-anal tail is absent. Phylum Chordata is divided into three subphyla: Urochordata or Tunicata, Cephalochordata and Vertebrata. Subphyla Urochordata and Cephalochordata are often referred to as protochordates in bracket figure 4.17 and are exclusively marine. In Urochordata, notochord is present only in larval tail, while in Cephalochordata it extends from head to tail region and is persistent throughout their life. Examples: Urochordata, Acidea, Selpa, Doliolum. Cephalochordata, Branchiostoma in bracket Amphioxus or Lancelet. The members of subphylum Vertebrata possess notochord during the embryonic period. The notochord is replaced by a cartilaginous or bony vertebral column in the adult. Thus, all vertebrates are chordates, but all chordates are not vertebrates. Besides the basic chordate characters, vertebrates have a ventral muscular heart with 2, 3 or 4 chambers, kidneys for excretion and osmoregulation, and paired appendages which may be fins or limbs. Figure 4.17, Acidea. The subphylum Vertebrata is further divided as follows: Vertebrata division, Agnatha in bracket leg jaw, Gnatostomata in bracket beard jaw. Agnatha class Cyclostomata, Gnatostomata in bracket beard jaw, divided into superclass Pisces and Tetrapoda. Pisces in bracket bear fins, Tetrapoda in bracket bear limbs. Pisces further divided into class 1 Chondrichythus and second is Osteichthythus. Tetrapoda further divided into class 1 Amphibia, 2 Reptilia, 3 Apes, 4 Mammals. 4.2.11.1 Class Cyclostomata. All living members of class Cyclostomata are ectoparasites on some fishes. They have an elongated body bearing 6 to 15 pairs of gill slits for respiration. Cyclostomes have a sucking and circular mouth without jaws. In bracket figure 4.18, their body is devoid of skulls and paired fins. Cranium and vertebral column are cartilaginous. Circulation is of closed type. Cyclostomes are marine but migrate for spawning to fresh water After spawning, within a few days, they die. Their larvae, after metamorphosis, return to the ocean. Examples: Petromyzon in bracket lamprey and Myxins in bracket hagfish. Figure 4.18, a jawless vertebrate, Petromyzin. 4.2.11.2, class Chondrichthyes. They are marine animals with streamlined body and have cartilaginous endoskeleton. In bracket figure 4.19, mouth is located ventrally. Notochord is persistent throughout life. Gill slits are separate and without operculum. In bracket gill cover. The skin is tough, containing minute placoid scales. Teeth are modified placoid scales which are backwardly directed. Their jaws are very powerful. Animals are predaceous. Due to the absence of air bladder, they have to swim constantly to avoid sinking. Heart is two-chambered, in bracket one auricle and one ventricle. Some of them have electric organ, in bracket example torpedo, and some possess poison sting, in bracket in bracket example trigon. They are cold-blooded, in bracket Poikilothermus animals, that is, they lack the capacity to regulate their body temperature. Sexes are separate. In males, pelvic fins bear clasper. They have internal fertilization and many of them are viviparous. Example: Scalodon in bracket dogfish, Pristis in bracket sawfish, Carcharodon in bracket great white shark, Trigon in bracket stingray. Figure 4.19, examples of cartilaginous fishes: A. Scalodon, B. Pristis. 4.2.11.3, class Ostreichthyes. It includes both marine and freshwater fishes with bony endoskeleton. Their body is streamlined, mouth is mostly terminal. In bracket figure 4.20, they have 4 pairs of gills which are covered by an operculum on each side. Skin is covered with cycloid/ctenoid scales. Air bladder is present which regulates buoyancy. Heart is two-chambered, in bracket one auricle and one ventricle. They are cold-blooded animals. Sexes are separate. Fertilization is usually external. They are mostly oviparous and development is direct. Examples: marine in bracket eggs. Examples: marine Exocetus in bracket flying fish, Hippocampus in bracket seahorse, freshwater Labio in bracket rohu, Catla in bracket catla, Clarius in bracket magur, Aquarium beta in bracket fighting fish, Pterophyllum in bracket angelfish. Figure 4.20, examples of bony fishes: A. Hippocampus, B. Catla. 2.11.4, class Amphibia. As the name indicates, in bracket group amphi, dual bios life. Amphibians can live in aquatic as well as terrestrial habitats. In bracket figure 4.21, most of them have 2 pairs of limbs. Body is divisible into head and trunk. Tail may be present in some. The amphibian skin is moist. In bracket without scales. The eyes have eyelids. A tympanum represents the ear. Alimentary canal, urinary bladder and reproductive tracts open into a common chambers called cloaca, which opens to the exterior. Respiration is by gills, lungs and through skin. The heart is 3-chambered, in bracket 2 auricles and 1 ventricle. These are cold-blooded animals. Sexes are separate. Fertilization is external. They are oviparous and development is indirect. Example: buffo, toad, rana, frog, hyla, tree frog, salamandra, in bracket salamander, ichthyofish, in bracket limbless amphibia. Figure 4.21, examples of amphibia: A. Salamandra, B. Rana. 4.2.11.5 Class Reptilia. The class name refers to their creeping or crawling mode of locomotion, in bracket Latin repair or reptum, to creep or crawl. They are mostly terrestrial animals and their body is covered by dry and cornified skin. Epidermal scales or scutes, in bracket figure 4.22. They do not have external ear openings. Tympanum represent ear. Limbs when present are two pairs. Heart is usually 3-chambered but 4-chambered in crocodiles. Reptiles are poikilotherms. Snakes and lizards shed their scales as skin cast. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Example: Chelone in bracket turtle, Testudo in bracket tortoise, Chameleon in bracket tree lizard, Calotes in bracket garden lizard, Crocodilus in bracket crocodile, Alligator in bracket alligator, Hemidactylus in bracket ball lizard, Poisonous snake, Naja in bracket cobra, Bangrus in bracket crate, Vipera in bracket viper. Figure 4.22 Reptiles: A. Chameleon, B. Crocodilus, C. Chelone and D. Naja. 4.2.11.6 Class Aves. The characteristic features of aves in bracket birds are the presence of feathers, and most of them can fly except flightless birds. In bracket example ostrich. They possess beak, in bracket figure 4.23. The forelimbs are modified into wings. The hind limbs generally have skulls and are modified for walking, swimming or clasping the tree branches. Skin is dry without gland except the oil gland at the base of the tail. Endoskeletal is fully ossified, in bracket bony, and the long bones are hollow with air cavity, in bracket pneumatic. The digestive tract of birds has additional chambers, the crop and gizzard. Heart is completely 4-chambered. They are warm-blooded, in bracket homeothermous animals. That is, they are able to maintain a constant body temperature. Respiration is by lungs. Air sacs connected to lungs supplement respiration. Sexes are separate. Fertilization is internal. They are oviparous and development is direct. Examples: Corvus in bracket crow, Columba in bracket pigeon, Psittacula in bracket parrot, Struthio in bracket ostrich, Pavo in bracket peacock, Aptenodytes in bracket penguin, Neophron in bracket vulture. Figure 4.23 Some birds: A. Neophron, B. Struthio, C. Psittacula, D. Pavo. 4.2.11.7 Class Mammalia. They are found in a variety of habitats: polar ice caps, deserts, mountains, forest, grassland and dark caves. Some of them have adapted to fly or live in water. The most unique mammalian characteristics is the presence of milk producing glands, in bracket mammary glands, by which the young ones are nourished. They have 2 pairs of limbs adapted for walking, running, climbing, burrowing, swimming or flying, in bracket figure 4.24. The skin of mammals is unique in possessing hair. External ear or pinnae are present. Different types of teeth are present in the jaws. Heart is 4-chambered. They are homeothermous. Respiration is by lungs. Sexes are separate and fertilization is internal. They are viviparous with few exceptions and development is direct. Examples: oviparous, ornithorhynchus in bracket platypus, viviparous, macropus in bracket kangaroo, pteropus in bracket flying fox, camelus in bracket camel, macaca in bracket monkey, ratus in bracket rat, canis in bracket dog, felis in bracket cat, elephas in bracket elephant, equus in bracket horse, delphinus in bracket common dolphin, Balanoptera in bracket blue whale. Panthera tigris in bracket tiger, Panthera leo in bracket lion. Figure 4.24 some mammals. A. Ornithorhynchus, B. Macropus, C. Pteropus and D. Balanoptera. The salient distinguishing features of all phyla under animal kingdom is comprehensively given in the table 4.2. Table 4.2 salient features of different phyla in the animal kingdom. Phylum Porifera, level of organization cellular, symmetry various, coelom absent, segmentation absent, digestive system absent, circulatory system absent, respiratory system absent. Distinctive features body with pores and canal in walls. Phylum Coelenterata in bracket Cnidaria, level of organization tissue, symmetry radial, coelom absent, segmentation absent, digestive system incomplete, circulatory system absent, respiratory system absent. Distinctive features cnidoblast present. Phylum Ctenophora, level of organization tissue, symmetry radial, coelom absent, segmentation absent, digestive system incomplete, circulatory system absent, respiratory system absent. Distinctive features comb plates for locomotion. Phylum Platyhelminthes, organ, level of organ. Phylum Platyhelminthes, level of organization organ and organ system, symmetry bilateral, coelom absent, segmentation absent, digestive system incomplete, circulatory system absent, respiratory system absent. Distinctive features flat body suckers. Phylum Aschelhelminthes, level of organization organ system, symmetry bilateral, coelom pseudocoelomate, segmentation absent, digestive system complete, circulatory system absent, respiratory system absent. Distinctive features often worm shaped, elongated. Phylum Annelida, level of organization organ system, symmetry bilateral, coelom coelomate, segmentation present, digestive system complete, circulatory system present, respiratory system absent. Distinctive features body segmentation like ring. Phylum Arthropoda, level of organization organ system, symmetry bilateral, coelom coelomate, segmentation present, distinctive system complete, circulatory system present, respiratory system present. Distinctive features exoskeleton of cuticle jointed appendages. Phylum Mollusca, level of organization organ system, symmetry bilateral, coelom coelomate, segmentation absent, digestive system complete, circulatory system present, respiratory system present. Distinctive features external skeletal of cells usually present. Phylum Echinodermata, level of organization organ system, symmetry radial, coelom coelomate, segmentation absent, digestive system complete, circulatory system present, respiratory system present. Distinctive features water vascular system radial symmetry. Phylum Hemichordata, level of organization organ system, symmetry bilateral, coelom coelomate, segmentation absent, digestive system complete, circulatory system present, respiratory system present. Distinctive features worm like with proboscis collar and trunk. Phylum Chordata, level of organization organ system, symmetry bilateral, coelom coelomate, segmentation present, digestive system complete, circulatory system present, respiratory system present. Distinctive features notochord, dorsal hollow nerve cord, gill slits with limbs or fins. Table end. Summary. The basic fundamental features such as level of organization, symmetry, cell organization, coelom, segmentation, notochord, etc. have enabled us to broadly classify the animal kingdom. Besides the fundamental features, there are many other distinctive characters which are specific for each phyla or class. Porifera include multicellular animals which exhibit cellular level of organization and have characteristic flagellated conocytes. The coelentrates have tentacles and bear cnidoblast. They are mostly aquatic, sessile or free floating. The ctenophores are marine animals with comb plates. The platyhelminthes have flat body and exhibit bilateral symmetry. The parasitic forms show distinct sucker and hooks. Aschelhelminthes are pseudocoelomates and include parasitic as well as non-parasitic roundworms. Annelids are metamerically segmented animals with a true coelom. The arthropods are the most abundant group of animals characterized by the presence of jointed appendages. The molluscs have a soft body surrounded by an external calcareous cells. The body is covered with external skeletal made up of chitin. The echinoderms possess a spiny skin. Their most distinctive features is the presence of water vascular system. The hemichordates are a small group of worm-like marine animals. They have a cylindrical body with proboscis, collar and trunk. Phylum Chordata include animal with possess a notochord. Other phylum Chordata includes animals which possess a notochord either throughout or during early embryonic life. Other common features observed in the chordates are the dorsal hollow nerve cord and paired pharyngeal gill slits. Some of the vertebrates do not possess jaws (Agnatha), whereas most of them possess jaws (Gnathostomata). Agnatha is represented by the class Cyclostomata. They are most primitive chordates and are ectoparasites on fishes. Gnathostomata has two superclasses, Pisces and Tetrapoda. Classes Chondrichthyes and Ostreichthyes bear fins for locomotion and are grouped under Pisces. The Chondrichthyes are fishes with cartilaginous endoskeleton and are marine. Classes Amphibia, Reptilia, Apes and Mammalia have two pairs of limbs and are thus grouped under Tetrapoda. The amphibians have adapted to live both on land and water. Reptiles are characterized by the presence of dry and the amphibians have adapted to live both on land and water. Reptiles are characterized by the presence of dry and cornified skin. Limbs are absent in snakes. Fishes, amphibians and reptiles are poikilothermous (cold-blooded). Apes are warm-blooded animals with feathers on their bodies and forelimbs modified into wings for flying. Hind limbs are adapted for walking, swimming, perching or clasping. The unique features of mammals are the presence of mammary glands and hairs on the skin. They commonly exhibit viviparity.